So in the previous class, we started with the chapter which is coordination of cellular fate processes, and we understood that there are three ways through which the cells can coordinate their cellular fate processes. Means they coordinate through cell-cell communication. And what are the three main ways of cell communication? First is cell secretes some soluble signals like growth factors, okay, and then these growth factors diffuse to a nearby cells, okay, and then they bind to suitable receptors on the cell membrane of the target cell and pass on the signal. Another way is via ECM, cells can also communicate, and lastly, through direct cell-cell contact, the cells can pass on few messenger molecules. And we started understanding the first mode of cell-cell communication, that is via soluble signals. We saw the types of growth factors, the cytokines and chemokines, right? We also saw a few examples of them and the roles of cytokine and chemokine. And we also understood. Oh, wait a second. Two people are in the lobby. Right. And we also started understanding the paracrine signaling, okay, like how in paracrine signaling the cell type which is producing the signaling molecule, the signaling cell, is trying to pass on the molecules to the target cell or the receiving cell. So with this figure we ended our last session, where this is the signaling cell, and from since the signaling cell is the source of the growth factor, so at the point here the concentration of the growth factor is the highest, and as the distance from the signaling cell is increasing, the concentration of the growth factor is decreasing exponentially, okay, and at some distance the target cell is there, and to its cell receptors the signaling molecules will bind. So we started with one question, that how far can soluble signals propagate. So we'll start today's class from here only, okay. So in this diagram we need to understand to how much distance this growth factor concentration is sufficient, okay, is sufficient for the target cell to respond to the signal. Means, or you can say, what is the optimum distance at which the target cell should be there with respect to the signaling cell, so that it can respond to the incoming signal. So that is the question basically. So let's try to understand and try to give an answer to this question. The maximum signaling distance, it can be estimated from a simple diffusion model describing the secretion from a sphere. So we will take help from the diffusion model. So here, since the growth factors are getting diffused, so choosing a diffusion model makes sense. Wait a second. Is Janvi not there in the meeting? Acha, let me. Okay. All right. So sorry for the delay, Janvi. So is any one of you is in touch with Kirtan? Apparently he is facing some issue with joining the meeting.
Yes, ma'am.
Yeah. So he is facing some login issue. Just help him out, okay? All right. So as I was saying that we are taking help from the diffusion process, because here the growth factors, they are basically diffusing. Here passive diffusion is going on from a region of high concentration to a region of low concentration. So, and we are considering cell as a sphere, which is the source of the signaling molecule. So, under steady state condition means we are understanding that there is no change in the uh, concentration with time, concentration of the growth factors with time. So, under that condition, we can say that the concentration of the secreted molecule is basically a function of the distance from the cell. So this is the concentration of the secreted molecule, and this is the distance that is from the cell. So we can correlate these two terms. Concentration can be correlated or with the distance under steady state condition by taking help of the diffusion model. So when you do that, you will get an expression like this, where C by Kd is equals to A into R by R, where C is the concentration of the secreted growth factor molecule, Kd is basically the dissociation constant, and if you do reciprocal of Kd, you will get the affinity constant. So it basically tells you at what strength the signaling molecule can bind to the receptors on the target cell. It is basically trying to quantify that affinity or that strength, whereas A is basically another constant, and it is a ratio of two time constants, time constant for diffusion and time constant for secretion. Here diffusion of the growth factors and secretion of the growth factors, and it is given by r square by d. Here capital R is the radius of the cell. We are assuming that the cell is a sphere, all right. So that is why a constant radius r is taken, whereas small r is the distance from the signaling cell, okay. That is small r. So it's a variable term. Here C is also a variable term. KD, A and capital R, these are all constants. So if any question given to you related to this, then you would know for what quantity to solve. All right. So, and here F, what is F? F is the secretion rate. At what rate the growth factors are getting secreted by the signaling cell. So yeah, that is F. Now, if we should estimate the distance that a signal reaches when C is equals to Kd. Here, what this condition means? This condition means that when a time point has arrived where the concentration of the growth factor released by the signaling cell is equivalent to how much of these growth factors molecule can go ahead and bind with the receptors. When these two things become equivalent, okay, then you will get a critical distance. Means at that particular distance you will get an optimal level of signaling going on in the target cell. All right. So in this figure, if the target cell is at a distance of r critical, all right, then you can expect that the concentration of the growth factors secreted by the signaling cell and the affinity of these growth factor molecules towards binding with the receptors, they are equivalent, and then you can say that maximum achievable signaling is possible. All right. So under this condition you can estimate that distance and at which maximum achievable signaling is possible at r critical. So you have to just put this uh, C is equals to Kd in this expression. Then what you will get? You will get R critical. It will go in the numerator, right? And A into R. And when you put the values of A, so you will get something like this. Or you can just simply put A. R critical by capital R is equals to A. Got it? And here A is nothing but a ratio of two time constants for the growth factors to diffuse away from the signaling cell, and the time constant for the secretion of these growth factors. All right. Now, let's try to understand another question. How long does it take to propagate a signal? All right. So, what is the, if you were to answer this question, at till what is the time required for the target cell to respond to a particular signal? It is another way of saying the same thing, like how long does it take to signal to propagate, or how long does it take for the target cell to respond? It's the same thing. Now, since it is an infinite amount of time, it takes an infinite amount of time to reach steady state. So this question cannot be answered under steady state. Steady state is an ideal condition that we uh, make in order to solve the problem easily. That is not a real life problem. Okay, that doesn't happen in reality. There is no steady state in reality. So a reasonable estimate of signal propagation distance is the distance at which signal strength is half of the maximum. So whatever equations that we saw in the previous discussion, they were all done under steady state. But here now the argument is in the steady state is like cannot happen, or it will take infinite amount of time. So we will estimate, or we will reasonably estimate that when the signal strength or the concentration of the signaling molecule is half its maximum, then that should be the signal propagation distance at which the target cell can respond. Now this leads to a time constant estimate for the signaling process of about 20 minutes and a maximum distance of 200 microns. So when you take this assumption, then you will get these values, that it will take 20 minutes for the target cell to respond, and it should be at a distance of 200 microns to respond, because at 200 microns the concentration of the signaling molecule will be half of its original secreted value. Okay. How strong can soluble signals be? Another very crucial question. So the nature of diffusion process is such that the highest concentration of the signaling molecule is found at the surface. Understood, right? And they decay in inversely proportional fashion with the distance of the signaling cell. As shown by this particular graph, as the distance is increasing, the concentration is declining in a inverse proportional manner, and it is declining exponentially. Correct? The concentration at the surface signaling cell is determined by substituting R is equals to capital R in equation 1. So this was our equation 1, and if you were to know what is the concentration of the signaling molecule at the cell surface, so you have to just substitute small r with capital R, alright? So you will get the maximum concentration of signaling molecule from that expression. So you will get C surface is equals to this value. Again, you have to do some calculation. Basically, substitute the value of A here. This is the value of A. You have to substitute here. Then you will get this particular value. Now it is said, and now this expression, what is saying that this will be less than or equal to the Rf max. What is F? F is the secretion rate of the signaling molecule. So F max will be the maximum achievable level of secretion, or maximum available rate of secretion. So obviously this will be the highest value that will give you the maximum concentration of the signaling molecule. Otherwise, at other rates of signaling, other rates of secretion, you will get a value like this. Now for effective autocrine signaling, autocrine means the cell is signaling itself. So for that to happen, the concentration of the growth factor at the surface should be greater than Kd. Obviously, the growth factors should more strongly bind to the receptors. All right, it is not should it should not be other ways, like Kd should not be greater than C surface. Otherwise, the growth factor molecules cannot bind. They have a more tendency to dissociate. they have a lower affinity towards the receptors. So estimates suggest that the value of sea surface is maximally about 1 nanomolar. So note these figures, all right? The times and the figures, the distances, the numbers basically. This will help you understand the complexity of this particular process that is going on at the cellular level. So if there are any queries, please stop me in the middle, all right? Because I may forget to ask you after every slide. So if you have any queries, then you guys let me know. So I was saying that estimates means people have mathematically estimated that the cell's concentration of signaling molecule at the surface is about 1 nanogram. So nanomolar. That is 10 to the power minus 9. That is so less. And this concentration actually limits the possible signal strength achievable with soluble signaling. So it is too less. It is too less for achieving the maximum possible signal strength in the target cell. So how can we tackle this? If we want to increase the signal strength in the target cell, how we do that? So we can do it by localizing the soluble molecules. Means we can localize it, means we can allow them to locally bind to some sites in the ECM, all right, so that they build up a stationary signal. So that signaling molecules, they remains stationary. They do not have the tendency to diffuse away. We are grounding those signaling molecules to state at that particular place. And place means we are building up a higher concentration of signaling molecules around the target cell by allowing them to bind to some particular sites in the ECM. So in this way, we can actually increase the signal strength in the target cell. All right. So this was all about how a cell sends a signal. All right. And the few major questions like how far it can go, how strong the signal can be. So these questions that we have already answered. Now the next question is, what happens when the signal is received by the target cell? Okay. So are there any queries so far? Yes, no? The equations that I explained, were they clear to you guys?
Yes, ma'am. Was
Uh,
clear.
okay, great. So the next question, as I said, is that we have to understand what happens at the target cell when the signaling molecule binds to the receptor. So there is a ligand receptor interaction going on. The growth factors, they bind to the receptors that are found in the cellular membranes and with a high affinity. So their association constants. So there was dissociation constant. The opposite of dissociation constant is association constant. So these values can be as low as 10 to 100 picomolar. So these values are very less. And we can actually describe the binding of a growth factor to a receptor using this type of an expression, where the binding is reversible as shown by this particular symbol. Means the GR, which is the growth factor and receptor complex, it can go back and individually form the receptor and the growth factor. Means they can dissociate from one another. And from this we can get the value of this dissociation constant, Kd. Kd is a dissociation constant, but Kd is equals to Gr by Gr complex. Now this type of expression, I'm sure you guys have seen in as equilibrium constants. Like in chemistry, we study equilibrium constants, where there is a reaction and we have to calculate equilibrium constants for that particular reaction. So if you recall that concept, you will see that it is written in exactly the same way. Okay. So Now the total number of receptors, okay, that is there in the cell is constant. So we will use the mass conservation balance, right? So our total will be basically the free receptors that are there are plus the receptors that have formed a complex with the growth factors. So it is a sum which gives the total number of receptors in the cell. Assuming that the binding is at equilibrium. Equilibrium means equal number of molecules have bound to the receptors, and also equal number of unbinding of receptors and the growth factors is going on. So equilibrium has been reached. So equilibrium and steady state those are totally different things, by the way. Don't confuse between these two terms. Equilibrium is different, steady state is different. In equilibrium there is no driving force, all right. So the both the events, that is binding of growth factors and unbinding, both have reached in a reached reached both are happening at an equal pace. There is no event that is favoring the one that is binding, or there is no event that is favoring only unbinding. Okay, so both have reached a balance, that is equilibrium. So if we consider that the bindings has reached an equilibrium, then we can write using this expression, this Kd. So you can then guess that this Kd is basically analogous to equilibrium constant here. All right. So from this Kd expression, we can substitute the value of R basically. Here R total is equals to, this value of R will come from this expression. So you can write R in terms of Kd, Gr and G, and then simply you just have to take out Gr to one side, and you will get an expression like this. So when you're asked that how many of the total receptors are occupied, that is basically the receptor occupancy. So if you were asked that, you will just simply get a ratio, that is the complex growth factor receptor complex that is formed divided by the total number of receptors. So this R total is there from this expression. GR is there on the right hand side. You just have to take R total to right hand side, you have to bring it down, and this whole thing, this particular Kd by G plus 1, this will go in the left hand side, and you will get this expression. Now people have experimentally got the values of the receptor occupancy. So this receptor occupancy is nothing but a fraction, all right. So you can understand, right? So this is going to be a fraction. So for the receptor of EGF, epithelial growth factor, okay, so epidermal growth factor, sorry. So this EGF receptor has an occupancy, or needs an occupancy of 0.25 or 0.5, that is 25% or 50% occupancy is required for significant stimulation, means for signal propagation. If 50% or 25% of these EGF receptor is not occupied, then the target cell will not respond to the incoming signal. Okay, so this is an example for EGF receptors. I'm sure in literature they must have reported for other receptors as well. So therefore, the growth factor concentration as low as 10 picomolar at the surface of the receiving cell can be sufficient to generate cellular response. Okay, so that is the final statement, that even if see, the cell we saw in the previous slide, right? At the cell surface, the concentration of the growth factor is around 1 nanomolar, right? 10 to the power minus 9. And due to diffusion, as it is diffusing away from the signaling cell and reaching the target cell, if that signaling molecule reaches at a concentration of 10 picomolar, 10 to the power minus 12, means 3 orders of magnitude of decrease is going on. Okay, then that is sufficient for generating a cellular response in the target cell. Okay, because this much concentration can occupy an approximately 25 to 50% of the receptors that is there on the cell surface. Now, the growth factors, when they reach the target cell, they are consumed. Or you can say they are endocytosed. Okay. Now, in many cases, the receptor ligand complex is internalized, means a vesicle forms around it, and the vesicle containing the receptor and ligand complex, it comes inside into the cytosol of the cell. And when that happens, the signaling molecule, it dissociates from the receptor, and the receptor is recycled back and then exposed on the cell membrane, whereas the signaling molecule, it is usually destroyed. So typical time constant for internalization and recycling to the cell membrane is in the order of 15 to 30 minutes. So it takes 15 to 30 minutes for the cell to first internalize and then recycle that particular receptor to the cell membrane. The absolute values for both factors, uptake rates, they have been measured. Means what is the uptake rate? Uptake means internalizing rate. Okay, so they people have measured for example IL-3, interleukin-3, SCF. This is also a growth factor. So these two growth factors, they are consumed by the hematopoietic cells, hematopoietic stem cells basically, at a rate of 100, so 10 to 100 nanogram per million cells per day. Okay, per day, per million cells, they consume this much of signaling molecule, or you can say 10,000 to 70,000 growth factor molecules are internalized. So these are some numbers on how much of the growth factor molecules are consumed by the target cell. So this process of receiving signal is thus dynamic, and the target cell may consume and destroy the signal. Means its task is not to pass on the signal to the other cell. Its only job was to take in the signal and do the intended function or start a downstream events. Okay, means show a cellular response. So since the signal is being destroyed, signaling molecule is destroyed by the target cell. So if the signaling cell wishes to uh, communicate with large number of target cell, then what will it do? It will have to provide a continuous flux of growth factors. Means it has to continuously produce signaling molecules over a period of time. Then only it can elicit enough cellular response in enough number of target cell. Okay. So any queries so far from anyone? All right. Now what happens when, so till now we were discussing or mainly focusing on the paracrine signaling. Means there is a signaling cell and there's a target cell. All right, and they are at some distance from each other. We are not going to touch upon the endocrine signaling, but we will see what happens during autocrine signaling. How does a cell sends as well as receives the signal? Means how does a cell signals itself, or what condition is required, or what people have observed that promotes, or what conditions that people have observed that promotes autocrine signaling. So autocrine signaling regulation, it has been implicated in pathological situation as well as in normal physiological mechanisms. So it happens during both in normal condition as well as in diseased condition. So for autocrine signaling, let's assume a ratio called A. So A is nothing but a ratio of the rate of ligand production by the rate of receptor production. Means how much of the or what is the rate or ligand production per unit time by the cell divided by receptor production per unit time by the cell. So when you do a ratio, you will get a value, and let's say that value is A. If the value of A is higher, okay, that means what? There is a higher rate of production of ligand in comparison to receptor, then there will be lower fraction of secreted ligand recaptured by the secreting cell. Means higher the value of A, higher the value of ligand production in comparison to receptor production. So there is not enough receptors exposed on the cell membrane, because of which there will be less formation of growth factor receptor complexes. All right. Conversely, in opposite sense, we can say the secreting cell almost completely captures the secreted ligand if A value is less than or equal to 0.3. Okay, so if this value, A value, is around this 0.3, then people have seen that the autocrine signal is happening at a maximum, because the secreting cell is able to recapture all the secreted ligands and form receptor growth factor complexes. Experimental analysis suggests that a very small A value elicits a significant systemic response. So A value should be very less, means there should be less ligand production in comparison to the receptor production. Then only people have seen that the cell is able to produce a large systemic response. If A approaches unity, means if these two production rates are equivalent, then most of the ligand will be lost to surrounding medium, and because of which the autocrine signaling strength will be less. There can be negligible amounts of ligand in the surrounding medium, yet there will be noticeable system effects. So this is what we can conclude, or we can understand from these statements, that during autocrine signaling, the lesser the number of ligands that is getting produced per unit time, okay, the easier for the cell to capture it, okay, recapture it using the receptors. So the signaling is happening and we cannot even notice it, because how we are noticing the signaling, whether it is going or not, we are measuring the concentration of the secreted growth factor molecules, correct? So autocrine signaling, it has been seen happening in many types of cells, like the mammary cells, prostate epithelial cells, as well as it plays an important role in regeneration of the liver and the proliferation of the immune cells. So in these different scenarios, we will have observed autocrine signaling taking place, and autocrine signaling happens in such a way that it is not even noticeable to us, okay, but the cell is responding to it very nicely, and we can see the systemic effect, means what changes had happened to the cell, we can observe that, but we cannot even see the ligands getting produced. It is almost negligible amount of ligands that are getting produced. So this is how the autocrine signaling takes place. So any queries, I would be happy to take up if there are any. No. All right. So maybe today's content is slightly on the lighter note, I think. Yes, no?
Yes,
Any?
ma'am.
Yes, all right. I was just going to think that nobody is there in the meeting because there is a pin drop silence. Now, let's also understand that how does a cell senses a concentration gradient of the soluble signal. So remember, for effective signaling process to happen, the cell can only respond to it when it senses a concentration gradient of signaling molecule, okay. So there should be a region of high concentration and a low concentration, and the cell senses it first. Then only if it is supposed to migrate, then it will migrate towards the region of high concentration, means towards the source of the signaling molecule, from which signaling molecule has secreted, if it is a chemotactic kind of a signal. And there are obviously other signaling as well, that is meant for simulating cell division or cell proliferation. Then in that case, growth factor concentration, I think, won't play a very major role. But during migration, cell migration, there should be a concentration gradient, okay, of the signaling molecule, that is chemokine that I'm talking about. Now cell is about the size of 10 microns. So this is an average cell size value. So we usually take 10 to 20 micron cell size. The chemotactic migratory cell may have to sense a gradient of chemokine to properly respond to the signal. Now given the cell size is very small and the concentration of the signaling molecule is also very less, means sometimes less than 1000 signaling molecule reaches the target cell. So it might be very difficult and sometimes impossible for the cell to sense a concentration gradient across its body. So what it does to overcome this problem, it usually extends lamellopodias. Okay, we saw during understanding cell migration process, right? The cell extends its membrane to form lamellopodias, and as it does so, it is trying to determine a concentration gradient, means trying to understand in which direction it has to move. Okay. Now signal has been received by the target cell, then it has to process. Okay, it has to undergo processing. What happens here? Now the binding of a growth factor to a receptor, it triggers a complex signal transduction process. So note, whatever events that is happening inside the cell after the ligand has bound to the receptor is quite complex, and active research is going on in order to understand the various biochemical pathways related to signal transduction process. And this process is very coordinated, and it consists of interconnected processes of signal detection, transmission and amplification. So this detection, transmission, amplification, these terms has been taken from circuit diagrams and all, where people study electric signals and all, but here they mean slightly different. Signal detection means the binding of growth factor to the receptor, means the cell is trying to detect, right? So detect it through receptors on its cell membrane. Then once receptor and growth factor complex is formed, then it has to transmit. How it will transmit? It will transmit through a series of secondary messengers that are there in the cytosol. Okay, and then there is amplification. So if the signal is supposed to switch on a transcription process or switch on the activity of particular type of enzymes, etc., then it will happen rapidly, means more and more molecules will pass on the same signal to each other and increase the rates of, say, transcription or translation, or enzyme activity of more number of enzyme molecules will increase. So this kind of amplification process will take place inside the cell. So once the signal is transduced, it will trigger a particular type of biochemical response. Okay. So biochemical response, you understand, some biochemical pathway will begin. So let's take an example in order to understand how signal processing is happening inside the cell. We will discuss only one pathway, that is interferon gamma signaling pathway. Has any one of you have studied this particular pathway? Means it, I think it is usually taught during bachelor's degree in cell biology, etc., kind of a subject. Has any one of you studied this particular pathway before? Interferon gamma signaling?
Not interferon gamma, but Jack's pathway, ma'am. Jack-STAT pathway.
Yes. So actually, that is what we'll discuss. Interferon gamma, when binds to its receptor, it switches on a series of jacks and stats, and then dimerization of stats happen, and then transcription of a gene takes place. So that is what we are going to discuss now. So first, the interferon gamma, which is signaling molecule, it binds to its receptor, which leads to dimerization of the receptors. So this is the, let me zoom in. So this is the cell membrane. All right, this is inside cell membrane, this is cytosol. So this is the receptors that are present individually. Okay, once the interferon gamma molecule, this is the interferon gamma molecule, comes and binds to one of the receptor, it will dimerize. So dimeric interferon gamma receptor will form. Okay, so then what happens? This dimerization then activates a series of components that is present in cytosol, that is called JAKs, Janus kinase tyrosine kinases. So these are basically kinases whose function is to add a phosphate group. So this dimerization activates these kinases. They will go and bind to the receptor. Okay. So these are JAK1, JAK2. These are went and got binded with the now dimer form of receptor. So these are kinases. So what they will do, they will add a phosphate group to the receptor. So these are the phosphate group. This phosphate group is coming from the ATP molecules. Right. Now this phosphate group, they have become anchor sites. Anchor sites for another type of molecule, that is STATs, signal transducers and activators of transcriptions. So these are again present in cytosol, and they bind to the phosphorylated receptors, and once they get binded to the receptor, they themselves get phosphorylated. So because of this, so they got phosphorylated once they got bound with the phosphorylated receptors. So due to this, they form the dimer. So this is stat dimer. Now this will act as a transcription factor. Okay, and it will go inside the nucleus and start the and it will bind to the promoter site of a gene and start the transcription process of a particular type of a gene. So this is an overall a very brief explanation of interferon gamma signaling pathway, which happens via the JAKs and STATs proteins that are there in the cell membrane. So sorry, cell cytosol. So I hope this was clear.
Ma'am,
Or yes.
please explain last step of uh, Jack pathway.
Okay. So here, what is happening? Note in this figure. So this is the receptor, interferon gamma receptor. Initially, it is present as single receptors. Okay. So once this interferon gamma molecules, they have come and formed a complex with this receptor. The receptor formed a dimer. They came together and formed a dimer. And then what happened? The JAKs, Janus kinases. Okay, so these are kinases. These are enzymes that can add phosphate groups. They also get activated upon the dimerization of this receptor. Once these JAKs got activated, they will go and get binded with the dimer form of the receptor. So this is a circle, and the rectangular part, these are the JAK1 and JAK2. They are now binded to the receptor. They will then add phosphate group to the receptor. These phosphate groups are the docking sites for another type of cytosol protein, that is STATs. Now these STATs will go and bind to the receptor, and this JAK will also phosphorylate this STATs molecule. So you can see that in STATs molecule there is one phosphate group. Now because of this, they are now activated due to phosphorylation. They are now activated, because of which they have formed a dimer. STATs dimer is formed. So now this STAT dimer form is the active form to function as a transcription factor. So it goes ahead and enters the nucleus, where it finds an appropriate promoter. This is gap promoter. It has bound to the gap promoter, and as soon as it binds, it starts the transcription of a gene. Okay, so once this gene will get transcribed, then translated and form a protein. This protein will become part of some biochemical pathway, and you will see some overall change in the cellular response. Is it clear now?
Yes, ma'am.
All right. So this example was help you, or was given to you to help you understand how a soluble signal, gets processed inside the cell. Now you must understand this signaling process is a very complex process, and often multiple signaling pathways, they are interconnected with one another. All right, but how do they interact? How these multiple type of signaling pathways interact with each other? So this interaction is happening in such a way that they precisely control all sort of fate processes, whether it's cell migration, division, differentiation, etc. All these are happening in a precise manner. So you can visualize this interconnected nature of the signaling pathways as circuits, interconnected circuits. So in 4 or 5 ways you can understand how the signaling pathway functions. So if this is a cell, this circular one is a cell, then you will allow few signaling molecule to bind to the receptor, means you give some input and you will get some output, means a cellular response. In the end you will get some cellular response. This is a very simple thing to understand, right? This is what we have been talking about. All right, in another case what will happen is multiple type of inputs, means multiple type of signaling molecules will come and bind to the receptor of the cell, whereas it will generate only one overall cell response, means there is only one type of output. Means although multiple types of signaling molecules are coming, binding to the different types of receptors, but all these signaling pathways, they are merging into producing only one cellular response. This is another type of understanding how these pathways are interconnected. Then another type, what will happen, this is a cell, a few signals, they are coming. Okay, few signaling molecules are coming. The cell is itself producing its own signaling molecules, which will go out, that will diffuse away from the cell, and there is few signaling molecules which are going and binding to its own receptors. So autocrine signaling also happening. The cell is responding to few signals itself, and at the same time the cell is secreting its own signaling molecules, and which are diffusing away from the cell. So this is how all these three types of processes are happening inside the cell at the same time. Where in the last figure here it is shown that there are few types of signaling molecules like P1 plus, P2, P3, P4, P5, P6. These signaling molecules, they are having a positive effect on the cell. Say it is a survival signal, means it is allowing the cell to survive, or it is a signal which is allowing the cell to divide. So these are positive effects which cell is experiencing. Whereas minus P1, minus P2, these are those signals which are negative, which are going to which are going to cause some negative effects. Like say it is going to remove some important crucial signaling survival signals. Okay, so or it is not allowing the expression of a particular gene that is crucial for the survival. So these are negative. So it is also a wonderful way of understanding how a particular cell is balancing the negative input as well as the positive input in order to survive. So in these four ways, you can understand how the multiple types of signaling pathways are interconnected with each other, and cell gives different types of responses. Now, now we have understood about signaling molecules, that is the growth factors. Now you should also know that the receptors, they are not just present in membrane bound form. Okay, they can be also present as soluble receptor molecules. So how these soluble receptor molecules are formed? Usually during the transcription of the receptor gene, what can happen? Alternate splicing can happen. So you recall the structure of a gene, like there is a promoter region, operator region, and there can be gene 1, gene 2, gene 3. Sorry, not gene 1, exons and introns. The sequence of exons and introns are there in the mRNA, remember? So during splicing, what happens? The what will remain? Exon regions will remain or intron regions will remain during splicing? Anyone?
Exons.
Correct.
Intron.
So exons regions will remain, whereas the splicing complex will remove the intron regions. So exon, there can be 3 exons or 4 exons, 1, 2, 3, 4. Now, if during splicing, the sequence of exon is changed, say instead of 1, 2, 3, 4, there is 2, 3, 4, 1. Okay, so this, due to this, another type of mRNA, mature mRNA, that will code for completely different type of protein, that will get formed. Similarly, 3214. So this is another sequence of this is another mRNA sequence that will code for another type of a protein. But all these proteins, they are coming from one gene. Agreed? So this is called alternate splicing. So when this kind of alternate splicing happens for a receptor gene, then it can generate two types of receptors. One receptor that is a membrane bound form, means it will go and bind to the membrane, whereas another type of receptor which is soluble in nature, means it will be secreted by the cell into the surrounding medium. Okay, so in this way, two types of receptors can be produced by the cell. Now we will discuss the responses, how like what kind of response that people have observed with respect to when cell is producing the soluble growth factor receptors. So mechanism A, here what is happening in this particular figure, a protease enzyme has come and cleaved the membrane bound receptor in left hand side. As a result, this growth factor receptor molecule, it has become a soluble form, means it will go and diffuse into the surrounding medium, because of which there will be no signal. So in this way, the membrane bound receptor can convert into soluble form, and when it does, there will be no propagation of any signal. The mechanism B, here what is happening, that there in the surrounding, that there is a large number of growth factor molecules. Now the cell is also producing a soluble form of a particular receptors. Means there are two receptor form, membrane bound and soluble form. And both the receptors, they have affinity to bind with the same kind of ligand. So the soluble form will try to then compete with the membrane bound form, because of which it will not allow the membrane bound receptor to pass on the signal. So this, if basically if soluble receptor is hindering, here is hindering by competing with the membrane bound form and not allowing, or have modified the cellular response, which it could have showed if the ligand was able to bind with the membrane bound form. So they are basically acting as inhibitors of signal propagation. In mechanism C, what is happening is the soluble receptors, they are acting as helpers. Okay, so since they are soluble, they can freely float the surrounding medium, and once the signal molecule has arrived, it is able to bind with it easily in comparison to the membrane bound receptors. So as it does, it is acting as a helper by bringing in the ligand molecule close to the membrane bound form, and because of which the membrane bound form can now easily bind to the ligand molecule and propagate the signal. So here the soluble receptor is preventing any harm to the ligand molecules, means it is preventing it from degradation. So there are other ways, right, that since it's a very small molecule, it can get degraded or it can be consumed by other cells. So the soluble growth factor here is acting as a helper and accelerating the process of signal propagation. When mechanism D, here what is happening, the membrane bound form and the soluble receptor form, they are dependent one another. The membrane bound form do not have binding sites, all right, so because of which when the ligands are coming, it cannot bind, whereas the soluble receptor, it has the binding site. So when the soluble receptor has successfully bound with the ligand, it is then associated. So here the soluble form by itself cannot propagate the signal, and the membrane bound form, since it doesn't have the binding site, so it also cannot propagate the signal. So if only they are able to work together, so only if they do work together, a signal will be propagated. Okay, and there is another mechanism where if and only if signaling will happen if the soluble form and the membrane bound form, they are synthesized together. Okay, and together they are transported to the cell membrane. As it happens, if it happens like this, the cell will be able to respond to the incoming signal. So a very good example comes from the CSF, that is the colony stimulating factor of granulocyte and macrophages, GM-CSF. This is a growth factor molecule. So the receptor for this growth factor molecule, alpha receptor and beta receptors. So these are two types of receptors for this particular growth factor molecule. Alpha receptor is the soluble form, whereas the beta receptor is the membrane bound form. So if these two receptors, alpha and beta, are synthesized together intercellularly inside the cell, then only they can respond to this particular growth factor, CSF growth factor. If somehow researchers have tested that if they provided the alpha form externally, like from the outside, if the alpha form is coming, then it will not bind to the membrane bound receptor, and thus it will not respond to the incoming ligands and propagate a signal. So here these two, membrane bound form and the soluble form, they are like brothers. They won't act alone. They have to be synthesized together, and then only they will act or respond to a incoming signal. So just with this slide, I'll finish today's class. So there can be, people have reported that there can be various kind of malfunctions that can take place during soluble signaling. So many of these examples have come from the cancer research, where people have seen, due to improper signaling, soluble signaling, cellular fate processes, they are not coordinated in the proper fashion, and that leads to the formation of a tumor. So a very good example of this malfunctioning comes from the studying this PDGF and the cis oncogene. PDGF is platelet-derived growth factor, which is a growth factor synthesized by the fibroblasts. So these growth factors, excuse me, yeah. So these growth factors, PDGF molecule and cis oncogene, these are very much similar to each other structurally. PDGF consists of two subunits, uh, basically two A chains or two B chains, or one A chain or one B chain. Whereas the protein synthesize by transcribing and translating the cis oncogene, people have found there is a sequence similarity between the cis oncogene protein and the PDGF PDGF molecule, especially the B chain. The B chain of the PDGF molecule, sequentially it is very much similar to the cis oncogene. So what happens normally in the cell, if the cell is expressing the PDGF receptor, so and it is expressing PDGF receptor, and the cell is also secreting PDGF molecule, so this molecule will go and bind to the PDGF receptor and do autocrine signaling. But at the same time, cis oncogene is also getting expressed in the cell. So what will happen, the receptor, PDGF receptor, will mistakenly bind with the cis oncogene molecule that is secreted, and because of which there will be abnormal activities. Means since it's a growth factor that is aimed at uh, stimulating cell division, so once the cis oncogene molecule, which is having the similar sequence, similar protein sequence as the B chain of the PDGF molecule, the cell will start dividing, and this will lead to uncontrolled cell division, and ultimately a tumor formation will take place. So this is a very simple example in order to understand how if there is malfunctioning in the soluble signaling, difficult to treat diseases like cancer can develop. So that's it from my end for today's class. If you guys have any queries, then let me know. I'll be happy to take them. Are there any queries?
No, ma'am.
Okay. So I hope today's class was interesting for many of you, because there was less maths and more biology, which you are familiar with, correct?
Yes, ma'am.
All right. So we have two more ways of signaling, cell signaling pathways that we need to understand. That is via how ECM, the cell is able to respond or communicate among each other, and how direct cell-cell signaling is happening. So we will cover up those topics in tomorrow's class, and and by tomorrow we will finish up the entire chapter of coordination of cellular fate processes. So let me just quickly take attendance. Wait a second. So is any one of you living in UP currently?
Yes, ma'am.
So how is the weather there?
Yes, ma'am.
Ma'am, it is rainy yesterday, but it is okay today.
So it here in Kanpur it has been raining for past 3 days. I don't know, and because of which many people I saw have cough and cold, and I think I may have contracted as well.
Yes, ma'am. The weather is too bad from Meerut and the West UP side, but it in the north side like Ghaziabad and the Shalgatan side, it is okay.
Okay, so one by one, tell me your name so that I can mark it.
Janvi.
Janvi, Vaishnavi, right?
Nidhi,
Vani,
Murli, ma'am.
Nidhi Priya, Murli. Anyone else? Sorry, I couldn't hear her. Shambhavi. Shambhavi, okay. So I have marked for Murli, Vaishnavi, Shambhavi, Janvi, Vani, and Nidhipriya. Okay. So there are only 6 of you today. Fine. So thank you guys for joining. I'll share the relevant content by tonight, and I'll see you guys tomorrow from 7 to 8 p.m. Okay. And then on Saturday morning, so Saturday evening, I think. Sorry, Saturday evening we'll have 1 hour quiz. Okay. So, all right, you guys can leave. If you have any queries, you guys can discuss with me tomorrow in tomorrow's class. Okay.
Bye, ma'am.